In Chapter 11, we're going to be discussing the efferent division of the central nervous system, and we'll be talking about the differences between our autonomic and somatic motor neurons. Please follow along in the study guide over Chapter 11. It'll cover this material. So our efferent division of the peripheral nervous system are the neurons that carry information from our central nervous system to the target tissues in the rest of the body. And it's made up of two types of neurons. Somatic motor neurons, which go to our skeletal muscles and they control our movement. Those neurons are largely voluntary, meaning that we are conscious and we have the central nervous system has perception and is directing those movements, and we're aware of those movements. Then we have the autonomic neurons. The autonomic division is being utilized by the body to maintain homeostasis and that dynamic balance that we talked about at the beginning of the semester. And so these neurons, they control our smooth and cardiac muscle, as well as a lot of our endocrine and exocrine glands, our lymph tissue, and even some of our adipose fat tissue. And most of these neurons are going to direct responses that are involuntary, meaning we don't perceive them, we're not aware of these things occurring. The two branches that make up the autonomic division are going to be our sympathetic and our parasympathetic neurons. So the sympathetic nervous system is associated with that fight or flight response. These direct those types of responses that would cause us to run away or to fight when we're confronted with a particular stimulus. You can think about like your heart rate increasing in speed. Someone jumps out and screams boo, and you feel your heart rate increase. That's the sympathetic nervous system that's elevating that fight or flight response. The parasympathetic, sometimes you can remember that by remembering the notion of rest and digest. These branches of the autonomic nervous system are targeting tissues, smooth and cardiac muscle, and causing us to engage in functions associated with either digestion or resting, like minimizing. So again, in the case of heart rate, if you start meditating after you've gotten scared, the heart rate is going to decrease. That's because the parasympathetic nervous system is now being stimulated and overriding what was happening on the sympathetic. And you can think of these two systems in large part being antagonistic to each other. So like this picture displays this kind of like seesaw scenario in which usually when one is being applied, the other is not. Both of these systems also work with our endocrine and our behavioral state systems in the brain to maintain homeostasis of our physiological set points. Where are these autonomic responses being directed? Where are the integrating centers? Well, most of the integrating centers are going to be associated with the brain stem and some areas of the midbrain. Often we refer to these regions as our primitive brain or the reptilian brain, because again, from an evolutionary standpoint, this part of the brain was the first to develop because it maintains homeostatic control of all these other physiological points. It doesn't necessarily have perception per se, but it's controlling those reflex controls. So we have these integrating centers that we find in the hypothalamus, right, in the pons, and in the medulla that are associated with these autonomic systems. So things like our respiratory center, found in the medulla of when we inhale, blood pressure control is found in the pons. We have urinary bladder controlled in the pons. And then in the hypothalamus, our temperature control that'll be directed through both an endocrine response in the thyroid, like we learned about, as well as a sympathetic parasympathetic response. water balance, eating behaviors are all taking place there. And as I stated, these two systems are under what we call antagonistic control, meaning that one autonomic branch is going to be what we call excitatory, and the other branch is going to be inhibitory. Again, I gave this example previously, but let's go back to it and look at it. What are the two antagonistic branches going on? But if we look at the effector organ being our heart, then the sympathetic nervous system is going to increase heart rate. That's going to be the excitatory. The parasympathetic is going to slow our heart rate down. That's going to be the inhibitory branch. And most of our organs operate with this antagonistic control. There'll be a few examples in which there isn't necessarily antagonistic control, but for large in part, this is how it functions. Now let's talk about the differences between some of the anatomical parts of our autonomic neurons, of the parasympathetic and sympathetic. So what do they have in common? Well, all of our autonomic neurons are comprised of what we call a preganglionic neuron and a postganglionic neuron. And the synaptic, or the synapse between these two neurons, is found in what we call an autonomic ganglion. So typically we'll have a single preganglionic neuron come on, and then we'll have an autonomic ganglion, and we may have multiple postganglionic neurons, or maybe not, maybe there's a single, but this is a nerve cluster that we have right here. So we have differences between our sympathetic and parasympathetic in one, the point of origin in the central nervous system, and then also the location of where we find these autonomic ganglions. So this is a great figure from your book. What you can see, it shows some of the differences between our two parasympathetic and sympathetic points of origin. And what you'll notice is that in the sympathetic neurons, they all, for the most part, originate in either our thoracic or lumbar spinal cord. And then the ganglions are very close to the spinal cord. So we'd have it on either side, right? What that means is that we have very short preganglionic neurons. And then after the ganglion, we have these longer postganglionic neurons that then go to the effector organs. Okay, so we have these two lateral chains that run on either side of the vertebral column. And this is where we have all of our ganglions. Whereas in the parasympathetic, the origins either start in the brain stem itself, or we have a few that start down in our sacral vertebral column, and they have very long preganglionic neurons, and then the ganglions themselves are found on the target tissues. So they're found here, and then we have very short postganglionic neurons. We also have differences in the type of neurotransmitters that get released in our autonomic nervous system. So both sympathetic and parasympathetic neurons, the preganglionic neurons, release acetylcholine. So we have acetylcholine that gets released from our preganglionic neuron, and then it'll bind to receptors on the postganglionic neuron, and those receptors are nicotinic. So we have nicotinic receptors that bind our acetylcholine, and that triggers the postganglionic. But now here we have the differences. So in a sympathetic postganglionic neuron, what's going to be released is norepinephrine. So that's the neurotransmitter released. Whereas in the parasympathetic neuron, we're going to release, again, acetylcholine, but this time it'll bind to muscarinic receptors. And again, these names, nicotinic and muscarinic, comes from the agonists that function with these, so nicotine and muscarine. On the sympathetic target tissues, we'll have adrenergic receptors, and they'll either be alpha or beta, depending on the target tissue that we're talking about. Okay, you will be responsible for knowing what type of receptors may be found, and what type of neurotransmitters would be released. So you'll notice that in your quiz for over this section, as well as what you'll have on your test. Okay, so these receptors would be cholinergic, right? These receptors are going to be your adrenergic receptors. On the target tissues themselves, what we find is, the axon of our postganglionic neuron, it comes to the smooth muscle or cardiac muscle, and then they diffuse through that tissue. And instead of having like a single axon terminal, what we find is that they have these varicosities, in which there's these bubbles that contain secretory vesicles with our neurotransmitters that are spaced throughout. So the reason they have this is so that they can affect that tissue, that smooth muscle all across, and have equal reactions. We'll talk more about this when we talk about actual muscle tissue types and some of the differences between that. But know that they have a varicosity that's going to differ from our somatic motor neurons. The process by which we release is very similar to what we talked about when we talked about neurons in general, right? The action potential will move down the membrane of the axon. When it reaches the varicosity, it's going to trigger voltage-gated calcium channels to open. Calcium will enter into the cell. As that calcium enters in this case, we're looking at a postganglionic sympathetic neuron because we're releasing norepinephrine. That triggers the exocytosis of our neurotransmitter secretory vesicles. Norepinephrine gets released, and it's going to bind to the adrenergic receptors on the target cell. Then it's going to diffuse away, out of the synaptic clefts, or it's going to be transported back into the cell, and we can break down norepinephrine utilizing monoamine oxidase that's found in the mitochondria in the varicosities. So MAO is the abbreviation for monoamine oxidase, and it's either going to be put back into a secretory vesicle or it'll get broken down into its constituents by MAO. These tables from your textbook, so 11.1, 11.2, specifically 11.1, a great table that's going to show, the differences between these, what's the inactivation enzymes for, our two divisions. When on the effector organs, MAO is going to be norepinephrine. Acetylcholine esterase will be for our acetylcholine in our parasympathetic system, how we have either nicotinic, muscarinic, cholinergic, or alpha beta adrenergic. It kind of outlines the differences between these. As far as our properties of the adrenergic receptors, you're not responsible for knowing these right now. I put this in here though because we'll be talking about these as we go on the semester. So we'll see, our alphas are generally found in most of our sympathetic target tissues. We get the 2s in our gastrointestinal tract and pancreas. Everything else is usually alpha 1. Where we get betas, though, those are associated with our heart and our blood vessels. One other difference that I want to talk about prior to moving on to somatic motor neurons is we do have in the sympathetic nervous system, an example of a pre- and postganglionic neuron that's a little bit different, and that is the adrenal medulla. So the adrenal medulla is a gland. It's found on top of the kidney, right? So we have this adrenal gland, and it's made up of two types of tissue, right? So we have the adrenal cortex, which is the endocrine tissue that will produce epinephrine, right? And we have the adrenal medulla, which is it's made up of neural tissue. Okay, so the adrenal cortex, it's going to produce some other hormones that get secreted in the body. And then in the medulla, we're going to release a neurohormone, epinephrine. So what we find, while most sympathetic neurons have very short preganglionic neurons, this is the exception, right? We have a very long preganglionic sympathetic neuron that then synapses directly in the medulla of the adrenal gland, and that's therefore going to be our postganglionic neuron, is all of this tissue. So while we synapse and release, our acetylcholine, this tissue then will secrete the epinephrine neurohormone that will enter in the bloodstream in the kidney and be circulated. So this is kind of our one exception to our rules of how our sympathetic differs from the parasympathetic. Okay, so let's move to our other group. We've talked about autonomic neurons, how they're used for homeostasis, antagonistic control, the differences between our preganglionic, sympathetic, preganglionic, parasympathetic, and then how the postganglionic, sympathetic, and parasympathetic differ. The other set of neurons that make up the efferent division of our peripheral nervous system are the somatic motor neurons. These are the neurons that trigger our muscles, our skeletal muscles, to contract. How do they differ from autonomic neurons? Well, first of all, their origin is going to be in the central nervous system, and it's just a single neuron. We don't have a pre and a postganglionic. We have a single neuron. It's myelinated, and then when it terminates, so the axon terminals are going to have branches, and then we will have what they're called neuromuscular junctions. Let's look at what a neuromuscular junction is. So here's our somatic motor neuron, right? Myelinated. It then comes and it has these axon terminals that end on what we call a motor end plate of the skeletal muscle. That motor end plate is the only region. So you notice we don't have a varicosity. We're not wrapping throughout all this skeletal muscle tissue like we see in our autonomic neurons in the smooth or cardiac tissue. We have these grooves that make up the motor end plate, and here are all of our skeletal muscle fascicles. And in this region, we have this membrane that has, it has all these what were called T-tubules. They kind of innervate a little bit deeper into the muscle. And this is where we have all of our nicotinic acetylcholine receptors. This makes up the neuromuscular junction. So we have this single spot that we innervate, and we'll get the entire muscle fiber, or in this case, motor unit, to contract. That'll make a little bit more sense when we talk about muscles next week. So we release acetylcholine, right? That acetylcholine, what triggers the release is that calcium coming in from the depolarization, and we will then bind to these nicotinic receptors, and when we bind the acetylcholine there, we basically open the channels to which sodium can come into the cell and potassium can leave the cell. Now, next week, we're going to talk about skeletal muscles, and so we'll talk a lot more about somatic motor neurons. But at the end of your book, there's a great table that then compares somatic motor neuron to autonomic. And what's the difference? Like the neurotransmitters that get released, all somatic motor neurons release acetylcholine. All of the receptors are nicotinic. Okay, whereas in autonomic, depending on whether we're parasympathetic or sympathetic, we have different neurotransmitters being released at the target tissue. Okay, make sure you understand these, you know these differences, because a lot of the questions that'll come on your test from this section is going to be, do you understand these differences between it? Which neurotransmitters get released from pre-sympathetic or pre-ganglionic sympathetic neuron? Which neurotransmitters get released from post-ganglionic sympathetic neuron, and so on. Okay, so pay particular attention to these